Hey everyone, welcome to the Basics lecture on cellular respiration. You can think of respiration and photosynthesis as making up a single cycle. The high energy electrons in glucose are used to make ATP in the mitochondria and then placed on oxygen to make water. The low energy electrons on water are excited back to high energy in the chloroplast using light and then placed onto carbon to make glucose again. We're going to do the first half of the cycle today and then do the other half in our next lecture. Moving electrons from glucose to the more electronegative oxygen releases a lot of energy. The many steps in cellular respiration are used to control the reaction and to make ATP. We have an organelle to talk about, but before we begin the biochemistry, the mitochondrion is an organelle that has a double membrane, like the nucleus does. The outer membrane is smooth. The inner membrane has folds called cristae. There are two important fluid spaces in the mitochondrion. The very middle is called the matrix, and the fluid between the membranes is called the intermembrane space. Here's an overview of all the reactions. Go to Chapter 9 in your book now and fill in these boxes. There are three major steps in cellular respiration: glycolysis, citric acid cycle, and oxidative phosphorylation. But notice only glycolysis occurs outside of the mitochondria. In this class, we're not interested in memorizing all of the intermediates and all the enzymes. Instead, I want you to know these things about glycolysis. First, it occurs in the cytosol outside of the mitochondrion. Second, glucose has some electrons removed, and these electrons are placed on a shuttle that carries electrons called NAD+, and when the electrons are put on, it makes NADH. Some ATP is made by substrate level phosphorylation, and the end product of glycolysis, called pyruvate, is transported into the mitochondrion. That's what you need to know for glycolysis. Now, glycolysis is the first place where we see our electron shuttle NAD+. Here's the structure, although you don't need to memorize it. Just recognize that it has a positively charged form, shown here, and then once it picks up 2 electrons and 1 hydrogen from glucose, it's a reduced molecule and called NADH. These molecules will pick up electrons at several different steps and deliver them to the electron transport chain. Another interesting aspect of glycolysis is that it makes a small amount of ATP. Not very much, because it's just moving a phosphate or two off an intermediate and onto ADP. Because this uses a simple enzyme, it's called substrate level phosphorylation. Here's a small example from the middle of glycolysis. The breaking of this phosphate bond here provides an energy to place the phosphate onto ADP, which makes ATP, and an enzyme is required to make this reaction go. Now let's stop for a minute and go down the side road of fermentation. Fermentation is a two-step process that includes glycolysis and then a reversing of the reaction where the electrons that were removed from pyruvate are put right back on. This resulting molecule in mammals is called lactate. This should strike you as a strange thing to do. Why remove high-energy electrons from a molecule and then put the electrons right back onto the molecule that you took them off of? This is actually done in order to create free NAD+. This molecule right here. This allows glycolysis to continue because it needs NAD+ to run. We'll discuss this more in class, but I want you to remember this. Fermentation is not directly a response to low oxygen. It's actually a direct response to low NAD+. Okay, so now we finished that side road. What happens after glycolysis? Pyruvate from glycolysis is transported into the mitochondria to enter the citric acid cycle. Here are the things you should know about the citric acid cycle. Pyruvate, which is a 3-carbon sugar, is fully oxidized down to carbon dioxide. The electrons from this reaction of oxidation are placed on the shuttles NAD+, which we've met already, and FAD, which is a new shuttle that acts similarly, and this creates NADH and FADH2. And again, a little bit more ATP is produced by substrate level phosphorylation. This scary figure is just here to show you that different foods enter cellular respiration in different locations. You don't need to know the exact details, just know this. Only carbohydrates enter at the start of glycolysis here. Both proteins and fats are important fuels, but they enter the cycle later at either the end of glycolysis, as in fats, or even down into the citric acid cycle, as in proteins. Finally, the third step to oxidative phosphorylation is the big payoff phase. Here, all the electrons that we've taken from glycolysis and citric acid cycle are carried by shuttles into a special part of the mitochondria and put to use. Let's have a look at how that works. Oxidative phosphorylation occurs on proteins that are located on the inner membrane of the mitochondria. One side of the chain is exposed to the matrix, and the other side is facing the intermembrane space. The proteins are called the electron transport chain. In the first step, NADH deposits its high-energy electrons on the first protein of the chain, and the energy is released when these electrons are allowed to drop to a lower energy state. The energy released when the electrons drop is used to pump hydrogen, ions, or protons up into the intermembrane space. The next protein accepts electrons from FADH2. Again, the movement of these electrons pumps hydrogens into the intermembrane space. And then at the very end of the transport chain in this molecule, all of the electrons have become very low energy and are now allowed to bond to oxygen to form water. This is our low energy reduced molecule at the end of cellular respiration. All of the hydrogens are now up here in the intermembrane space, and they are allowed to fall from a high concentration to low concentration, but only through a molecule called ATP synthase, shown here. This particular protein changes shape when the protons run through it, and this conformational change provides the energy that places phosphates onto ADP and creates ATP. This is where the majority of ATP is made in cellular respiration. Now notice we've made ATP in two different ways, substrate level phosphorylation and glycolysis and citric acid cycle, and this process called chemiosmosis, which occurs in oxidative phosphorylation that uses the strength of the hydrogen ion concentration gradient to make ATP. The mechanism on this side is much more efficient, produces a lot more ATP, but you can see that it requires a whole factory of membranes and proteins and high pH in order to maintain this system. Finally, I don't want you to memorize how many ATP or how many NADHs are made at each step, but I do want you to recognize that the majority of ATP occurs via chemiosmosis rather than by substrate level phosphorylation. And this completes our basics video for today. Write out the answers to the following questions to study for the basics quiz. Thanks for watching.